Good afternoon, CGIL. This week, we're very happy to have Dr. Roberto Carvalheiro presenting for us. Dr. Roberto Carvalheiro is a principal research scientist in aquaculture applied breeding at CGIL in Australia. He holds a bachelor in animal science from the Federal University of Lavras and a master's and a PhD degree in animal breeding and genetics from São Paulo State University, UNESP, with part of his doctoral research conducted at the University of Guelph in Canada. With over 2 decades of experience, Dr. Carvalheiro worked extensively in animal breeding, quantitative genetics, and genomics. He has served as a consultant at Genesis in Brazil, where he played a key role in implementing genomic selection in commercial beef cattle breeding programs. He later joined UNESP as a senior researcher from 2013 to 2022, and since 2022, Dr. Carvalheiro has led the research at Cesario focused on applying genomic to commercial aquaculture breeding programs, collaborating closely with industry partners. His work aims to develop sustainable breeding strategies and translate complex data into practical solutions for livestock and aquaculture sectors. He has authored over 150 scientific publications, supervised numerous graduate students, and delivered more than 15 invited talks at academic, scientific, and industry events. So the floor is yours, Dr. Carvalheiro, and you can get started.
Thank you, Julia, for the introduction, and thank you very much for inviting me to give the seminar. It's a great pleasure for me to have this opportunity. And as I was talking to Flavio, thank you so much also for making the recordings of the previous talk available. It's a really great source of information. And that's, I think, is the first time I'm going to give a talk at 5:30 a.m. So I hope my brain works well. So let's see how it goes. So today I will talk about the power of genomics in aquaculture breeding. But before talking about the technical aspect, I just would like to make some acknowledgements to CGIL, especially to Flavio Schenkel, because of the great contribution CGIL had to my academic formation. I spent lots of different students coming from UNESP to University of Guelph. I was one of that students as well a long time ago, more than 20 years ago now, as a visiting PhD student, and it was great for my academic formation. I also had the chance and the great privilege to interact with researchers from the University of Guelph, with Flavio, with Steve Miller, with Mehdi Sagozé, and others. And this interaction was really helpful when I was working at the industry in Brazil, and helped a lot us to properly implement genomic selection in beef cattle breeding programs there. And also when I moved to the university in Brazil, great collaboration, especially with Flavio. He received 3 of my students as visiting PhD students as well. And it was very fruitful collaboration in terms of science and in terms of human resources as well. So thank you so much. Going to the technical part of the presentation, I split this talk in 3 main sections. I will discuss the power of genomics, genotypic information to optimize aquaculture breeding programs. And then we'll talk a little bit about the power of genomics to uncover unexplored source of variation, and also the power of genomics to bridge the gap we have between breeding nucleus population in aquatic species and commercial farms. So before we talk about some details about the benefit of using genotypic information in aquaculture breeding program, I'll just talk overall in terms of the breeding scheme. Genomic selection has not caused a great change still. There is the possibility of making some change in the breeding scheme. But usually, if you compare the left-hand side figure with the right-hand side figure, you will see great similarity in terms of breeding nucleus and seed testing and multiplication farms and things like that. So the use of genomic information it was just an additional component of collecting tissue of samples and of animals and genotyping them. So when we see this, we don't see big change here, But as we are going to see in the next few slides, genomics has allowed to optimize, really optimize these breeding programs. And in the next slides, we'll see, we'll discuss first about general aspects of the main components of traditional breeding programs, aquatic breeding programs. And then what was the benefit of incorporating this genomic information? In terms of the broodstock population, most aquatic species, in comparison to livestock species, the domestication is very recent. So usually what happens when breeding program, aquaculture breeding program is being established, usually the main source of broodstock is wild animals. So, but the choice of these animals and the selection of these animals, it's quite crucial because after a few years, usually because of the high fecundity of most of the species and the high genetic gain that's possible to achieve, and also due to biosecurity reasons, usually these breeding populations, they are closed after a few years of selection. So choosing properly the broodstock source is crucial. Also, because of having usually closed populations, it's quite important also as well to make a breeding scheme with a reasonable number of families being controlled. Here we are talking about usually 300 families per cycle. And it's quite important to use optimum contribution to control genetic diversity and inbreeding. Something that's quite important as well is to think about a strategy to mitigate the risk of losing broodstock. So usually having a backup population or having a cryobank, it's almost mandatory to mitigate this risk. How genotyping information has improved this component of the breeding program. So in many different aspects, here I'm listing some of them. As I mentioned, broodstock sources is quite important. So with genomic information, we can have a much better assessment of the genetic diversity of the founders population. So it's quite important, and it has helped a lot to make better designs and better schemes to make sure that we'll have enough genetic diversity in the breeding program. It has also allowed to improve the inbreeding control. Optimum contribution can work really well without genomic information, but the level of resolution we have to control inbreeding with genomic information is much, much better. We can, for example, identify regions in the genome where we are increasing a lot autozygosity. So, and we may check if there is any inbreeding depression effect in that region. Also, in terms of the backup population of the cryobank, we can instead of making use of pedigree information and trying to store different families in the cryobank, we are able to use, for example, haplotypes and increase haplotype diversity in the cryobank or in the backup population. So the level of resolution to make decisions is much better and improves the efficiency of the full breeding program. In terms of reproduction and family hearing, this is the main bottleneck for a wider adoption of selective breeding in aquaculture. And for some of the species, it's very, very hard to have full control of the full living cycle and the reproduction. So, and because of that, even for the species where we have the control of reproduction and life cycle, sometimes we select some broodstock candidates for reproduction, and some of them they are not ready to spawn. So that's common. So that's why usually we select a higher number of families that we need for family production, because some of the animals broodstock candidates, they don't respond to the reproduction protocols. And another thing in family production is that before genomics, we needed to have the families in different tanks before tagging the individuals. So that created a problem because for some traits there was an important confounding effect with tank and family effect. And because of the high fecundity of most of the aquatic species, an important aspect in this component of the breeding program as well is that we don't need, because every family produces lots and lots of progeny, lots of individuals, we don't need to keep all of them as breeding candidates. So we can, if we have a good strategy, we can do press selection here and within family. How genomics has allowed optimization of this component of the breeding program in different ways. Here I'm reporting, I'm giving some examples. I mentioned that in some case the broodstock candidates, they are not ready to spawn. So we may use genomic information, genotypes these candidates and calculate GBV for maturation or for readiness to spawn, for doing a pre-screening and having more predictive ability in terms of the broodstock that are going to reproduce. Another big advantage of using genomic information in this component here is that we can hear all the families. We can do communal hearing of the different families in the same tank if we want, because we are going to tag the individuals, to pit tag them, and to sample, to collect a sample of them to genotype, so we can do family assignment with high precision. So we don't need to wait to pit tag the animals before having them in the same tank. So that's a big advantage. Here in this component of the breeding program, it's probably the main benefit of using genotypic information comes from here, because without genotypic information, usually the selection of the breeding candidates, they were made using family EBVs. So we have limited intensity of selection here. And also in some species, it's very different, the environment in which the breeding candidates, they are heard in terms of in comparison with the commercial farms. In salmon, for example, the breeding candidates, they are in the breeding nucleus animals, they are in controlled conditions in fresh water. And commercial farms, the animals in the last part of the production, they are in the ocean. So it's quite different, and genotype by environment effect can be quite substantial. So there's lots of limitations here. And also when you do some challenge tests for some disease in SIP test conditions, because these tests, they are really usually quite expensive, and the level of accuracy for selection of those traits, if you don't use genomic information, you use only family EBVs, so you don't have good return over investment when you invest on these difficult to measure traits. With genomics, it's quite amazing because most of the breeding programs we do partnership to do research and development activities. They are really willing to incorporate new traits and these challenge tests. For example, the return over investment it's much bigger now because you can select the animals with much higher precision. We are migrating from family EBV to individual GBV, so you have much better accuracy of prediction. You can fully explore within family variation, so you increase selection intensity. So as I was mentioning, you have a much better return over investment for hard to measure traits. And as a consequence, you may have much more comprehensive selection index and more balanced selection. And also a great benefit of genomic information is when we think about the multiplication part of the breeding scheme. Here it's quite interesting because only you don't need to worry here about controlling genetic diversity because it's like a terminal cross. All the fish will go into commercial farms and then to the consumers. So you don't need to be worried about genetic diversity here. So you can do really high selection intensity here. But before genomics, this selection intensity was based on family EBVs, so we were not fully exploring within family variation. So we had a constraint on intensity of selection here, and in the accuracy of selection as well. And also limited, because of that, of having limited accuracy, we were not able before genomics to fully explore G/E and non-additive genetic effects. But with the incorporation of genomics, in theory, we can really redesign the breeding scheme and apply prediction equations to fully explore G/E non-additive effects. In theory, we can even use information coming from commercial farms using digital technologies, for example, to calculate genomic predictions based on these traits measured at the commercial level and feed the prediction equations in the breeding population. So this is still underexplored. There's a great opportunity here, and I guess this is where genomics is going to cause big change in the way we design the breeding scheme in aquaculture breeding nowadays. So just to summarize this first section of the talk, before genomics, communal rearing was possible only after tagging. So there was a confounding effect here between tank and family effect. Limited capacity to manage genetic diversity. Emphasis on selection for growth. Minor investment in hard to measure traits because of the low return over investment. So selection was predominantly based on family EBVs. It was constrained. We had constrained capacity to explore G/E and non-additive genetic effects, and it was very difficult to implement a tailored commercial multiplier production. But after genomics, we were able to do communal rearing of the individuals from different families before tagging. That's a great advantage. Even doing that, we are able to do precise family assignment with genomic information. We have improved our capacity to manage genetic diversity. Selection accounting for several economically relevant traits and selection based on individual GBVs with much high accuracy. And we also have improved capacity to explore G/E and non-additive genetic effects. And as I mentioned in the previous slide, we have opportunity to redesign the breeding scheme for tailored commercial productions in different environments and market conditions. One question that most of people always do, and it was a question mark I had when I started working with aquaculture as well. Is genomic selection cost effective? Because when you think about a value of a fish, you don't think it's worth investing in genotyping. But because the high fecundity of each one of these broodstock, minor gains in genetic gain we have in your breeding population, it's going to translating huge impact in the multiplication in commercial farms. So it's quite cost effective, actually, the investment in genotyping. Here, just to show some results in terms of relative increase in accuracy by using genomic selection from data. Very comprehensive review paper published by Ross Houston and co-authors, and they show that yeah, depends, of course, in the heritability of the trait, the number of the individuals you have in your reference population. But as for most of the species, the gain by using genomic information in the accuracy of prediction it's quite substantial. And in Atlantic salmon in Tasmania, for example, the gain of using genomic information for the selection index was estimated to be about 30%. Another thing that is quite interesting and can help us to save some money in applying genomic selection in aquaculture breeding, because we have large full SIB and half SIB families, and because the breeding candidates they are very closely related with usually the reference population used to build the prediction equations. Usually low density SNP arrays, they are quite effective. There is this study here showing that with few thousand of SNPs, you are able to, I'm hearing my voice back here. I'm not sure. Yeah, that's better now. Thanks, Julia. Yeah, with few thousand SNPs, it's possible to obtain the same level of accuracy of higher density SNP chips. Of course, using low density SNP chips, you can save some money, but there is a plateau. But we are really, that's a really strong area of research from our team. We are investing a lot of effort and energy here. And my colleague Kurtzlin has presented a very interesting talk in Spain at the International Symposium this year, showing for, especially for emerging species and for species that we don't have full control of the life cycle for example, or they are emerging and they don't have capacity to invest in this classical breeding program nucleus structure. So there are very interesting alternatives of implementing low-cost genomic selection in aquatic species, especially using DNA pooling. And that's not only theory. We have applied this concept in different programs, and one successful story is in Barramundi, where we helped the industry to establish a breeding program 6 years ago, and using low-cost genomic selection implementation, and the company was able to save almost 90% of the cost in genotyping, and is being able to obtain expressive genetic gains. So there are different alternatives there to implement genomics without big costs. The second part of this talk is to show the power of genomics to uncover an explored source of variation. And I'm going to present an example in Atlantic salmon. I'm just checking the time. Okay, we are good. So in Atlantic salmon, we are talking about the same species here, but it's quite interesting. There is the North American lineage and the European lineage, and they have different number of chromosomes. So aquatic species, they are quite interesting because they have many different things, and as background in livestock, I'm learning a lot to work with aquatic species. So in this case, the North American counterpart, it has 2 chromosomes less than the European one, mainly because of the 3 big rearrangements. There was the fusion. We are talking about very ancient things happening here in the chromosome. So the fusion between chromosome 8 and 29, the fusion between chromosome 26 and 28, and the fusion of chromosome 1 and the fusion of part of the chromosome 1 into chromosome 23. So there are 3 big chromosomal rearrangements here. And as we will see, it has important impact in some important traits. The main evidence of this was found in different papers investigating wild Canadian populations. So these papers, they show strong evidence that these chromosomal rearrangements, they act as supergenes and they contribute to local adaptation. For example, this first paper here, the frequency of fusion, it was related with summer precipitation. So there are different, and these other studies showing association with local conditions in different rivers as well. So we found this evidence, and then we decided to investigate if this structural chromosomal rearrangements, if they were segregating in Atlantic salmon in Tasmania as well. So salmon in Australia was imported from Nova Scotia, Canada in the 60s. So if there is evidence that these chromosomal rearrangements, they are polymorphic in Canada, we imagine they were polymorphic in Tasmanian Atlantic salmon as well. And we wanted to investigate their association with adaptive and production traits. And we found very strong evidence that these rearrangements are also segregating in Tasmanian Atlantic salmon. When we plotted the first principal components of the genomic relationship matrix, when we use all SNPs from all chromosomes, we can see this clear structure pattern splitting the Norwegian, the European, and the Canadian and the Tasmanian populations. They are very distinct. But when we target SNPs surrounding the fusion between chromosomes 8 and 29, we can see that they are in the North American, the Canadian, and the Tasmanian lineage. We have some individuals closer to the Norwegian, to the European cluster. And when we investigated this further, what we call as the group 1 here, which is the Tasmanian, which is apart from the Norwegian cluster, it has in the fusion region, it has very low minor allele frequency and very low level of heterozygosity. This is explained by when the fusion happens. Usually there is suppression of recombination in that region. So we were able to identify that this cluster corresponds to the fused chromosome or the North American karyotype, if you like. And the group 5, then it's the unfused. So the SNPs in chromosome 8 and 29, they segregate. The minor allele frequency and the heterozygosity level indicate they segregate independently. And the group 3 is the heterozygote karyotype. And interestingly, when we compared the difference in the genetic merit of these karyotype groups, we observe a strong association for some traits, some really important traits. For example, the North American karyotype was, in comparison to the European karyotype, it was superior for amoebic gill disease resistance, for maturation at freshwater and marine water, for summer performance, summer growth at high temperatures, and for harvest weight. The European karyotype was better than the North American karyotype for post-summer survival and for summer survival. What's really interesting, in addition to be able to be associated with these different traits, what's really interesting is that these rearrangements, they follow Mendelian inheritance patterns. So, for example, North American karyotypes, if you have broodstock with two siren dam with North American karyotypes, they'll produce only progeny with North American karyotype. European karyotype, the same. So it's possible to design the mating allocation to favor the prevalence of one karyotype or the other, depending on your breeding goals. The third section of the talk, we will discuss about the power of genomics to bridge the gap between nucleus and commercial populations, especially using DNA pooling strategy. So in here, the example is in salmon again. So what happens, the SIP testing, usually it's very similar conditions to the commercial farms, but there are some differences, and there's always this question: is the information we are using from the SIP tests and doing the selection, what's the impact in the real commercial and in the real farm production? So and we are using DNA pooling to answer that question, to validate the genomic prediction equations built in the commercial population. And we are also starting to use information coming from commercial farms, especially in terms of survival, to try to build good prediction equations at the commercial level and to select broodstock in the breeding nucleus. And I will show you some results about our investigation my colleague team did, and to investigate the genetic base of post-handling mortality in Atlantic salmon. It's a big problem because usually here we are talking about these tanks here. They have they have about 60,000, 70,000, sometimes 100,000 fish. And sometimes they need to handle those fish to do some management or to do some control. And if you have a fish almost close to the harvest and you have mortality here, it's a big loss. So we were trying to identify a good indicator traits measured in the breeding population to try to see if these we could help the industry to reduce this handling mortality at the commercial farms. And in the beginning, there was the people were suspecting that this could be related with cardiac traits. So the main hypothesis in the beginning of the study was to investigate if cardiac traits were associated or not with these post-handling mortality in commercial population. So the strategy used here, we estimated SNP effects from traits using data from progeny test or SIP test. And we started with cardiac traits, but because we had genomic prediction for production and other traits, we ended up investigating all the different associations. We collect tissue samples from commercial animals that died and survived after these handling events. We pull tissue samples by this binary trait, by this phenotype, dead or alive, and we extracted DNA and genotype. Then we calculated the GBVs of each pool, and compare the average GBVs of the mortality and survival for the different traits assessed in the breeding population. So in terms of the DNA pooling strategy, as I mentioned, tissue samples were pulled by this binary trait, by phenotype. We used 20 samples per pool, and randomization to remove cohort effect, because this handling mortality events, we evaluated in 10 different cohorts. And we had 50 pools per phenotype, representing a total of 2,000 individuals. DNA was extracted from pooled samples, and they were genotyped using a custom 50K SNP array. In terms of the cardiac traits, the only significant difference we found was for heart weight. So fish with higher heart weight tended to have higher mortality. But when we corrected the heart weight for the weight of the fish, that's the cardiosomatic index, the difference was not significant. So that means that actually it's not the heart weight. The problem is that fish that have higher weight, they are more prone to die. So that was the main. And it was not associated with heart shape or heart fat. In terms of the production traits, again, fish with a higher harvest weight tended to have higher mortality than fish with lower harvest weight. And for condition factor, the same. So this is indicating to the industry that possibly they need to be thinking about strategy to overcome that, because fish that is more interested for the industry in terms of weight, they tend to have higher mortality. So the industry needs to do something to control this indirect genetic response, undesired genetic response. And what's quite interesting is that we observe this summer survival was calculated using information from the SIB tests. And it showed that progeny from families that have higher summer survival also had higher survival at commercial farms. So indicating that summer survival at the progeny or SIB test, they can be used as an indirect trait for handling survival at commercial farms. Also, there is a significant association with post-summer survival and with growth during hot summer. So some concluding remarks. Genomic information has allowed to improve the efficiency of aquaculture in different ways. Some we have discussed here in this presentation. But in my point of view, its potential it's still largely underexplored. Genomics has allowed viable and cost-effective implementation of selective breeding programs in emerging species through customized or tailored breeding scheme. We have interesting example here in barramundi, for example. Genomics is assisting to uncover an unexplored source of variation for complex traits. And here I show one example of the chromosomal structural rearrangement in salmon. Genomics is helping to bridge the gap between breeding nucleus and commercial farms. Again, I showed an example here with salmon data. And the benefits of genomic information extend beyond these applications that we show here in this presentation. There are many other benefits that we can have with the use of this information. I started the presentation acknowledge my colleagues from the University of Guelph, and I would like to conclude my presentation acknowledging my colleagues from CSIRO. It's really an amazing team to work with. And here I'm also listed some colleagues from CSIRO from other teams who have a really great contribution in our work. Thank you for all the team, and thank you all for your attention. Back to you, Julia. Can you hear me or? Okay. I cannot hear you. I cannot hear you. I still cannot hear.
Roberto, can you hear me?
I can hear you.
Yeah, so I think it's something with the room there.
Yep.
But
I was
now,
at
now
some stage at my presentation, I was hearing the sound back from the room, and I think Julia fixed that, but now she's not able to.
Can you hear me now?
yes.
I can hear now. Yeah.
Okay, that's perfect. Yeah, we had a problem in the room, but now everything's working well. Thank you for
Yep.
understanding that.
No worries.
So we said thank you. So thank you very much for your presentation. I would like now to open the floor, either from those here in the room or those join us online.
I have a question.
We do have a question in the room. Yeah, just click, and when it's green, you can talk.
Press. Okay. No.
Green?
Yeah, it is.
Mhm.
Yeah. Hello, doctor. Can you hear me?
Yes, I can hear. I don't
Oh,
know who's
perfect.
speaking, but I can hear.
Yeah. My name's Bruno, and I also work with fish. Thank you for your presentation. On the part of reproduction and family rearing, it made me curious about the estimation of breeding values for maturation, because it is constant challenge with the breeding season, that you have some candidates and you do a pre-selection of them, but then a big part of them don't get to mature. But in that case, do you estimate breeding values and the pre-selection and incorporate it in the selection index, or you do it as a first step before applying the selection index? And in any of those cases, what is the estimated heritability for it? I know this may probably be with Atlantic salmon.
Thanks, Bruno, for the question. You can use both strategies, incorporating in your selection index and or as an independent trait, the pre-selection. So it's a heritable trait, so it's possible to do that. It depends on the breeding objectives of the industry and then the selection index. But because of the fecundity of the species, you can do without the necessity to incorporate in the selection index, unless there is, I would say, unless there is very strong negative correlation with the selection index and this trait, you need to think about alternative ways to handle this problem. But yeah, usually it's possible to first to record this information properly, and then to build a good prediction equation for that trait, and then to incorporate that in your breeding scheme, either using independent selection for this specific trait, or as you mentioned, by incorporating the selection index. But yes, it's heritable trait.
Thank you, doctor. And in that case, you mentioned the data, the phenotypes, and I'm curious about how they are like, how they are like recorded, and if you need, by your experience, how much data would you need to have, because you have a limited population of breeding candidates, and also you would have different year classes that you collect the data.
Yeah, there are different ways you can do that. For example, the trait can be time to maturation, where you expose your broodstock candidates to different inductions, for example, and record those that responded to the induction or not. Or you can, if you really want to push hard, for example, and you, for example, use as a binary trait females that were ready at in Atlantic salmon, for example, females that were ready at 2 years of age, or they were not ready. So, for example, so there are different ways you can determine the trait, and in terms of numbers, there's, as for all the other traits, there is no magic number. You need to start recording and starting assessing the quality of your prediction equation, doing cross-validation, and to check the level of predictive ability that the breeding program will be comfortable to use that information.
Yes, thank you, Doctor. And so you're proposing two scenarios, and if I understand correctly, in the first one, there's a hormonal management in the phenotyping, and in the second one is through the whole like breeding season, with recording it as a binary trait. And the management of maturation with, like, for example, hormonal treatment is something that's interesting, but many companies do not apply it. But maturation is one thing, but then the time in which a breeder achieves that maturation is variable, and sometimes there's not coordination between the best animals that you want to reproduce. So there are some technologies, like for example semen refrigeration, and I wanted to know if you have like looked into that experience, because I have been thinking about it for a company I work with, but it has some limitations because you have to propose a economically good proposal.
Yeah. I think we are entering here in many specific details. So I would be interested to know more about that, and but it's difficult to give a suggestion when someone makes you a question in the spot, because it's usually better to have a holistic view of the system, what's happening, to recommend a proper trait and a proper design to evaluate and to propose that trait. But yeah, I would be very interesting to talk more about that.
Yes, for sure. I will write to you, but from your experience, have you seen that it's very applied, like the refrigeration of semen there in aquaculture?
Yeah. As I mentioned, there is this cryobank strategy. So sometimes they use mute from the cryobank if they want to recover genetic diversity that is being lost or something like that. So it's possible.
Okay, thank you, Doctor. Those were my questions. Thank you.
Thank you, Bruno, for your questions. Any other questions, either from here in the room or those joining us online? I do have one question. For the multiplication. Flavia, please go ahead.
First of all, thank you, Roberto, for this presentation. It's always good to listen to people that are working other species that we don't work every day. So, but one thing that I always I think about in terms of aquaculture, right? we have our species, livestock species that we work, and we have, for instance, sheep, goats, that are very difficult to convince the producers in the industry that it pays off to genotype the animals, right? Because they say, well, it cost me $30 to genotype, and my ewe costs $300, $200, right? So relative to the what the animal values is hard to convince, right?
Yeah.
Different with like cattle in general. But then I think about fish, right? So can you tell me how much costs for the genotyping, right? And it gives me an idea of the value of a seed stock animal, right? Could be a male or a female that you have there. That's the seed stock, it's in the nucleus.
Yeah.
How you calculate this? No, I know that it pays off. Otherwise, the industry will not move towards genotyping, right, and use genomic information. But how is this the cost of genotyping with the how much worth the individuals, the seed stock?
Yeah. Yeah, so I will give some examples about numbers, but thanks for the question, Flavio. I will give some examples just for you to have an idea, but there are many different cases and many different scenarios. So I just give an example, but it's if the ability to convince is it's mainly related to the to the industry and to the scale of the operation, because for example, when you have I mean the business model, when you have the same company that has the broodstock, they have the commercial, the multiplier and the commercial farms, it's way easier to convince that genomics pays, it's cost effective, because they see the benefit of that clearly. And for example, if they invest in every cycle, if they genotype, let's say, between the breeding candidates and the SIP testing, if they genotype, let's say, 10,000, 20,000 animals per cycle, and we are talking about a cost around $20 per sample, we are talking about an investment of I mentioned my brain, it's early in the morning, my brain is not working well. Let's go back to the numbers. $20 per sample. If we talk about 10,000 animals, so we're talking about 200,000, or 20,000 animals, we are talking about $400,000. So some investment per cycle between $200,000 and $400,000. But when these broodstock, they produce the commercial fish and the fish that go to the farms, so the gain it's really huge. So and because you are able to fully do a selection intensity really strong and fully explore within family variation, so the benefit it's much higher than the investment.
Yeah. So Roberto, what is the ratio of multiplication, right, between a seed stock animal and the animal, the commercial animals? How many times that individual multiplies?
You know it's crazy, yeah. So yeah, each broodstock produces thousands of progeny. So
it's like in poultry, right?
Yeah.
And that's the problem. It's not the problem, but it's the structure of the breeding scheme, like to have this pyramid, but also the fertility, right?
Exactly. Yeah.
Right. The difference in fertility, right, is amazing in aquaculture. How many offspring you could have for a pair of individuals.
Yeah.
And that you don't have in this small livestock species like the mammals.
Yeah.
If you get sheep and goats, they could have these breeds that very common to have a triplets, for instance. Yeah. So then the one individual have a more impact.
Yeah.
But it's really it's a different game. It's very hard to convince them to genotype. But yeah, and another thing, I think difference is the genetic improvement, the rate of genetic improvement that you can achieve in aquaculture compared with any other species like land species.
Exactly.
Yeah, that's interesting. Thanks again, Roberto, for the presentation. And my last question is not related with the presentation, but are you going to be in the World Congress?
I hope so. I submitted an abstract. So let's see if they are going to accept it.
So I will see you there.
Okay.
It'll be great to see you again and talk to you personally there.
Yeah.
Thanks a lot. I will let others to ask questions.
Thank you, Flavio. Do we have any other questions? Yeah, we do. Please, Timothy.
Can you hear me?
Yep.
Awesome. So is the correlation between high harvest weight and mortality common among most aquaculture species? And is it a physiological reason or a disease load type?
Oh, that's a complex question. Thanks. Yeah, the biology behind that, we have still many question marks, and it's interesting because for the same data set that I gave the example, when we calculated the correlation between survival and harvest rate, we found post-summer survival and summer survival with harvest rate. We didn't find any genetic correlation. So we are still investigating that indirect genetic response. And I don't have a clear answer right now, but in different species, it's quite common to have these antagonic effects between weight and reproduction. For example, it's very common in some species when you do very strong selection for weight, you compromise reproduction, for example. So I guess maybe it's like an imbalance of energy or something like that. But we have much more questions than answers for the biology behind that.
Thank you. I appreciate it.
Thank you very much, Dr. Roberto. Thank you, Timothy. This has been a great discussion. Thank you again for presenting for us today, Dr. Roberto Carvalheiro. And thank you for waking up so early. We really appreciate that. Thank you, everyone, for coming. The recording will be posted on YouTube, and we will share more details about that with you later, Dr. Roberto. And see you next week. Have you all a nice weekend.
Okay. Thank you. Thank you, everybody. Bye-bye.
Thank you.